Hello, everyone. I'm Cherry Kubota, Ohio State University. Welcome to Indoor Ag Science Cafe. This is a June Cafe. And I appreciate you share your time, busy time in your schedule to be with us talking about technologies and science of indoor farming. So as usual, before I introduce today's speaker, I'd like to go quickly over some business stuff. So today's agenda, let's make it small so that's not on my way. Okay, so it's a presentation. We are going to have Dr. Erik Runkle from Michigan State University, and he's going to talk about selecting an LED fixture for indoor plant production. And then this part is going to be recorded. And then after that, we will stop recording, and then you can ask any questions, hard questions, easy questions. Erik can answer all the questions.
Yeah.
So anyway, all right. And conference, workshop, events information, I just put some of the things I know I'm involved here. Plant empowerment workshop, this is more for greenhouse, but I think the concept of balancing physics and plant physiology applies to indoor also. That's July 9th and 10th, organized by Ohio State. The second one is a Cultivate 13th to 16th virtual. And then the third one is Indoor Ag-Tech Innovation Summit. I haven't attended this one before, but I might be able to attend this year. So I wanted to share. I don't know if anyone participated. Erik, did you participate this meeting before? Yeah, it's going to be virtual again. So there are a lot of virtual conferences this year, which may be a good thing to test out, new conferences you've never been. And then the last thing, I think Dr. Runkle is going to talk more about the details, but we will be organizing as a project, OptimIA stakeholder meeting. Actually, this is gonna be online, I believe. But organization is from Michigan. All right, so, okay. Today is again,
Sherry,
yes, question?
That will also be virtual, correct?
Right, yes.
July 29th, okay.
That's correct, Chris, thank you. So this is June Cafe, right? And in July, usually we don't have cafe because that's a conference month, and then my schedule is crazy. So I just follow the tradition of not having cafe in July. Plus we have a stakeholder meeting instead, so that's good. August meeting, our cafe is gonna be given by Dr. Neil Matson from Cornell University, and then also Marianne Niemann from Rensselaer Polytech Institute, if I pronounce correct, RPI in New York. And then they are gonna provide quite interesting topic comparing nutritional quality between 3 distinctly different production systems, indoor, greenhouse, and outdoor. So I think it's a good topic. Everyone wanted to know, and this is one of the questions I constantly get asked. So with that, I like to stop sharing and then have Dr. Erik Runkle start sharing his presentation. Oh, that's right. I need to give a.
You need, yeah.
Yeah, you should be able to do now.
Yes, thank you.
Mm-hmm.
Well, thank you, Cherry. I chose a topic that I thought would be of broad interest, but I also think I've challenged myself because it doesn't relate to a specific experiment. It's basically a compilation of projects that we have done, as well as observations and understanding research that others have done. I think it's somewhat therefore subjective, and I certainly don't assume or pretend that I have the answers, but a lot of this is really sharing my insights, and I think some of these things are probably obvious, and other things may be things that you haven't really thought about. So what are the sorts of considerations when deciding what sort of fixture or fixtures to use for indoor plant production? And of course, indoor plant production can vary widely. It can span from micropropagation to ornamentals to specialty crops like cannabis to leafy greens. And it's going to be somewhat situational depending upon the crop and as well as the quality characteristics. Briefly, I want to acknowledge some of the major horticulture and lighting companies that have supported the research we have performed at Michigan State. This certainly is not an exhaustive one, but I would say it's the major contributors. And then a disclaimer there at the bottom that I'm going to be mentioning a few products, but they don't necessarily mean or imply an endorsement by me or Michigan State or bias against products or companies not mentioned. This is a photo that I took in Japan at QP Corporation. I believe Kerry Mitchell mentioned the company when he gave that interesting summary of the history of indoor farming. And you can see a photo I was able to take where they're growing lettuce under high pressure sodiums. And on the same visit, we also visited Chiba University, where they've done a lot of research with indoor farming. And at the time, LEDs were being used to a small extent in Japan, but still most companies were using fluorescent tubes. And I think that was the case until the beginning of this past decade. Of course, now operations pretty much exclusively use light emitting diodes. Companies have been doing this for several years now. One of the first installations I saw was in the Netherlands in 2011, and they were among the first to convert from fluorescent tubes to LEDs. All right, so sole source lighting is very important. Looking at a business facility or a business develops for indoor farming. I'm not an economist, but I know that the capital expenditures to purchase and install the LEDs as well as the operational costs for the lighting have a big influence on the profitability of indoor farming. Lighting is important because the daily light integral, or the accumulated amount of photosynthetic light per day, has a big effect on crop yield. And in fact, for every 1% increase in light, most crops, the harvestable yield of them increase by about 0.8 to 1%. So there's almost a 1% increase in yield with every 1% increase in light. The quality of lighting also regulates a lot of the quality characteristics, things like extension growth, leaf color, the nutrition, potentially even the taste and the texture of edible crops. It can also influence things like flowering and ornamentals. And then the efficiency, or the efficacy is the term we like to use, of an LED fixture is important because not only do the higher values mean that they consume less energy to deliver the light, but also less waste heat needs to be removed from the facility. So it's kind of a double whammy in terms of higher the value, less electricity needed for both lighting and for HVAC. These are considerations. I may have missed a few, but the major considerations when determining what sort of light fixture to use for indoor farming. The light intensity, or specifically the photosynthetic photon flux density, is important, and related to that, how many hours can you light plants during the day? The product of those two yields the daily light integral. The choice of a spectrum, and that's probably the most challenging aspect of determining what fixture to use in your facility. Is it a fixture that has a fixed spectrum, or is it one that can be adjusted in terms of the intensity or perhaps the photon spectrum? Related to that, what is the light quality? Is it blue and red LEDs? Is it composed of white and red, white, blue, and blue, red, and far red? There are lots of different permutations. And what are those effects on the yield of the crop as well as the quality? And we can't also forget the impact of lighting on people and our experience in an indoor farm. What is the cost of the lighting, not only for the fixtures, but the cost to install the fixtures? How uniform is that light delivered to the crop? Of course, what is the efficiency of the LEDs and the fixture itself? Is it a qualified horticultural product by the Design Light Consortium or the DLC? And then last but not necessarily least, what is the customer service of the company from which you're purchasing the fixtures, and does it have a positive reputation? And if they do have a warranty, which increasingly fixtures do, will they be in business if you do have problems down the road? These are just some examples of photosynthetic photon flux densities that you might see depending upon the crop and the stage of production. So micropropagation would be at the low end. You might see facilities with as little as 25 micromoles per meter squared in second, up to maybe 3 times that. And then depending upon how many hours of light you're delivering, you can see the DLI might be as low as 1.5 moles up to maybe 5 or 6 moles with a longer photo period. We've done a lot of work with ornamentals, and there are obviously going to be exceptions, but generally we're seeing or recommending intensities somewhere around 140 to 180 micromoles. And again, depending on the DLI, depending on the photo period, your DLI will be anywhere from about 8 to 13 moles. And then leafy greens, again, we see examples of less than 150 and more than 250, but a lot of companies are using intensities at this range, although I will say that it seems like companies keep pushing that number, and I've seen instances where we're getting up to 300 micromoles, and there may be examples of higher intensities. And so therefore you're getting daily light integrals of 10 up to 20 or maybe even more moles per day. So when coming up with light intensity, you need to consider what sort of crop are you growing, as well as what are the photoperiod limitations, if any. So if it's a photoperiodic crop, you may be limited in terms of the number of hours per day you can deliver light to the plants. For example, if it's a short day plant, you might want to give it a long day, but if you're trying to induce flowering, you would need to give it a short day. And then there are crops like tomato and some other solanaceous crops that start to develop intumescence when the photoperiod exceeds 18 to 20 hours. And as I mentioned before, the selection of the intensity also influences the demand of the HVAC system, because higher intensity, of course, there will be more waste heat associated with it. So there are 3 general types of fixtures available. Most LED fixtures for indoor lighting have a fixed photon spectrum and a fixed intensity. And these are the lowest cost, but they also have the least flexibility in a facility. Some fixtures are available that are dimmable. So the spectrum doesn't change, but you can change the intensity. That, of course, comes at a slightly higher cost, but it gives a little more flexibility in their use. And then there are other fixtures that were primarily for research applications, but they allow independent LED channel control. So you can change both the intensity and the spectrum of the photons emitted to the crops. So these are the most expensive but have the most flexibility. Carrie, I see some comments, but I'm not able to read them in this view. So if something is noteworthy, please interrupt me, okay? So these are some very general effects of different radiation wave bands on plant growth. Of course, there are going to be exceptions, there are going to be interactions, and there are going to be some effects of light intensity on these. But generally, we think of UV and blue radiation as promoting the color or increasing the coloration of leaves, increasing the nutrition of edibles, as well as increasing the leaf thickness. They also act to inhibit extension growth and can suppress crops that have intumescence, like some of the solanaceous crops. Green light is known to generally penetrate a canopy better than other wave bands. In some cases, it can promote extension growth, although we have also seen some instances where it can suppress extension growth. Green also, because it can increase extension growth, can decrease the coloration as well as the thickness. And then far red, which is an area that we've done a lot of research on, we know and others have shown that extension growth is promoted by far red. With long day plants especially, it can promote flowering, but because it promotes extension growth, it decreases leaf coloration as well as leaf thickness, similar to the effects of green light. Okay, so we've done a lot of research in the controlled environment lighting lab at Michigan State. And the research I'm going to show you is going to primarily be generated at Michigan State, but obviously many other researchers are performing and have performed and published a lot of interesting data. And so I'm just going to be showing a very small set of some of the research that we have done, looking at the effects of light quality, specifically on growth of leafy greens. Okay, so I want to share just a couple experiments. These were performed by Qingwu Meng when he was a PhD student at Michigan State. And they're basically two experiments that were designed to better understand how blue, green, and far red influence lettuce growth when added to a fixed photon flux density of red light. So some of the parameters we looked at were the yield, or the fresh mass of the plant, the leaf size and shape, as well as color. We did some research on nutrition and consumer preference, but I won't talk about that today. So I'm going to focus just on the growth attributes. So this is a somewhat of a control treatment where we have 120 micromoles of red with 60 micromoles of blue, a relatively high photon flux density of blue. And then we designed treatments that were modifying this. And so we kept red the same in all of, or most of these treatments. We have 120 of red. And then we partly or completely replaced some of the blue with green radiation. So you can see, for example, in this treatment, we have 20 micromoles of blue, 40 micromoles of green, and then the red is the same. Here then we are replacing some of the blue with far red radiation. So here we have 20 micromoles of blue, 40 micromoles of far red, and then still the 120 micromoles of red. And then we had three other treatments where we have partial replacement of the blue with green and far red. We have the warm white LED as a control, and I'd like to point out that the warm white LED for total photon flux density was the same in all of these treatments, and it included 10 micromoles of blue radiation. And then the last treatment was a mint white LED, also known as an equalized white, and that emitted about 15 micromoles of blue with just a little bit of far red. So I want to show a video that William took of 4 of these extreme treatments. So we have the control with the red and blue only, the red and green, the red and far red, or the warm white. And so this is a video that shows basically these lettuces growing under the different spectra at the same time, same temperature, everything is the same except for the light quality. And very quickly, within a few days, you can see effects on the shape or the architecture of the plant. Very compact growth under the red and blue in the upper left, and the opposite on the lower left where you have the red and far red radiation. And so again, I think this is all relatively well known, but I guess what struck me was just how quickly we see a response in these sort of treatments. And it also suggests that there may be opportunities or desire where you would have one spectrum to do one thing, and then as the plant matures, you might switch the spectrum to get different growth characteristics. This is a representative plant from each of the treatments when the plants were harvested. You can see the different treatments represented by the pie charts and the effects basically on the size and coloration of the different lettuces. So this is the red leaf lettuce Rouxai. You can see here the treatment is quite purple, relatively inhibited extension growth. As you replace some or all of the blue with green radiation, you can see the diameter increases, but the coloration of the leaf decreases. You see similar effects when you replace some of the blue with far red. And then you can see the controls with the warm white and the mint LED where there is kind of an intermediate coloration as well as leaf size. I want to show you some graphs then, and we thought about what's a good way to try to understand what the responses are. And there was a commonality in a lot of these responses with respect to the blue photon flux density. And so we're going to present different parameters, in this case leaf length, of the lettuce Rex, which is a green leaf variety, and the effects of blue photon flux density when we are partly or completely replacing blue with green. So as this graph shows, this is with leaf length. We see here this would be no blue, so we would have 120 micromoles of red and 60 micromoles of green. And then as you increase blue and decrease green, you can see the leaves become progressively shorter. We see very similar and even more pronounced effects with far red, where you have far red and no blue, you get even more extension growth, and that decreases as you increase the blue photon flux density. Very similar results with kale in terms of the trends, although just to note that in this case, the green light replacement with blue caused an increase in leaf area in this case, slightly more than, slightly more than the effects of far red, but relatively comparable. Now we look at shoot fresh mass. Again, these graphs are designed similarly, and we see that when you are basically increasing the blue and decreasing far red or decreasing green, you can see that you get an associated decrease in the fresh mass. Similar results, in fact, almost identical results with green and far red with the kale Siberian. And then what was interesting was we wanted to see if there was a correlation between leaf area and shoot fresh mass, because we see the lines and the shapes of these are very similar, whether we're looking at leaf area or leaf length and the shoot fresh or dry mass. Excuse me, I guess we're not presenting dry mass, this is all shoot fresh mass in this case. And so we can see that they're very well correlated. So as you increase the diameter of the plant or the leaf area, you can see that you're getting increases in the shoot fresh mass. So that suggests that a big reason for our growth responses and fresh mass responses can be attributed to the effect on extension growth and probably the plant's ability just to capture and use the radiation available. For more information, you can see a reference at the bottom, and if someone doesn't have access to that journal, then feel free to send me an email and I can send you a reprint. Okay, so one of the questions that came out of this, though, was to better understand, or desires was to better understand the effects of green radiation and how much of the effect was from green versus a decrease in the blue, because we were always changing the two at the same time. And so we developed a second experiment that tried to tackle this question. So in this case, all treatments had a total photon flux density of 180 micromoles. And then we started with that 180 and then started to substitute some of that red with blue radiation to the point where we had a very high intensity of the blue. Then we had treatments where we replaced some of the red with green radiation and then had similarly increasing photon flux densities of blue. So we have a case here, for example, where we have 100 micromoles of blue, 60 micromoles of green, and only 20 micromoles of red. And we had two additional treatments. One was a warm white at 180 micromoles as a control. And then we grew plants in the greenhouse that were under somewhat similar conditions with respect to temperature and daily light interval. Again, these are representative plants from each of the treatments at harvest, and this is for the red leaf lettuce Rouxai. Not surprisingly, we have a relatively large head of lettuce under only red, and as you go left to right, we see increasing blue, and the effect that has is on inhibiting extension growth, but also to some extent increase in the coloration. Here's the warm white control there on the right, and we see actually relatively similar responses as red with just a little bit of blue. And then if we have green and red, again, as we start to substitute some of the green with red and as we increase the amount of blue, we see very similar results as we saw with just the red and blue treatments. So if you compare these two, one above each other, you can see at least visibly, the plants are very similar. Looking at some data, again, after looking at the data and thinking of how do you present the information, we again saw very similar trends as we saw before, where whether or not we have green radiation in the photon spectrum, so this would represent data without green light and with green light, that the shoot fresh mass was highly influenced by the percentage or the total photon flux density of the blue radiation. So as we increased the amount of blue, we got less extension growth, and that resulted in less biomass production. And so that was the case with the fresh mass here, and here we have the shoot dry mass. Just to point out, this is the warm white LED, which again has about 10 micromoles in our treatments of blue radiation. And you can see that if you were to mark where that 10 micromoles was, it fits very nicely with the impact of blue driving the shoot fresh mass when the total photon flux density is kept constant. Okay, so jumping to some other parameters, obviously we think of light quality and the effects it has on plant growth. But let's not forget the effect it can have on people. This is a red and blue treatment that may be a very efficient LED fixture, but it can be a challenging environment in which to work. Contrast that with primarily a white LED, much more pleasant environment. And then this would be a fairly common spectrum that's maybe, we could call it a pinkish spectrum that has a fair amount of white, but also some red. And that's done for various reasons, but the result is that you can get a more pleasant environment, almost as good as the environment with just white. The fixture installation and costs, this is of course very important. To work with lighting companies or their suppliers to obtain a customized lighting map and price quotation for a specific light intensity that you request. So basically you would identify what is the photon flux density, probably specific sort of light spectrum or choices of light spectrum. And then they would work with you to tell you, okay, you need this number of fixtures and they should be spaced in this way. The quotation should be for your specific facility, your specific operation, and also considering the height from one canopy to another. So the distance from a lighting fixture to your crop. So they should be able to provide you with a lighting plan and details of the light spectrum, the efficiency of the fixtures, the expected lifetime of the fixtures, and then hopefully they offer some sort of warranty. Light uniformity is something that I think we're not as cognizant of as we should be, because it can have a big impact on the uniformity of the crop. There are some operations that are using sunlight as well as kind of a hybrid of a solar and indoor operation together. And so while sunlight is free, so to speak, the downside is you get tremendous differences in the uniformity of the light in terms of the spectrum and in terms of the intensity. And so if you were to measure the intensity in the spectrum throughout this area, it's going to vary from one location to another. And I think one of the major advantages of indoor growing is that we can take a lot of that uniformity out. We can take a lot of that variability out to create a much more uniform environment. So I think this is a very challenging case where your uniformity of lighting is quite poor. Indoors necessarily, though, you're not getting a better uniformity if lighting is delivered still as a point source. So this illustrates well where you have quite a high intensity in the middle, and it then will decrease rather rapidly as you get to the edges. And so you're going to have, of course, different architecture of the plant as you decrease the intensity, as well as changes in the biomass. This can be easier to manage if you're in a hydroponic system because the water demands for different crops can be met. But if you're growing in some sort of substrate that needs irrigation, the plants in the middle will certainly need more water than the plants at the edges. And so for these reasons, when you're completely in an indoor facility, and you have fixtures designed so that the spectrum and the intensity are uniform, the result is you'll have much more uniform crops. This is a chart published from a paper by Bruce Bugby's lab this year. The citation is at the bottom, and it's useful because it shows several things, but a couple things I want to point out is that the efficiency of an LED, or really the efficacy in terms of the number of photons emitted per unit of energy, is influenced by how hard you drive the fixtures, but more importantly, what is the type of LED. So you can see the efficiency or the efficacy is greatest for far red LEDs and slightly lower for red. Blue are lower, and of course the lowest of these are white. Now green is not in here, it's going to be much lower, and that's really why we don't have green LEDs in most cases for commercial operations. The efficiency is influenced by how hard it's driven, and so the harder you drive it, it's influenced by the current density, and it can also be influenced by the temperature. So when we're thinking about developing or using an LED fixture, we have to think about what is the composure of the LEDs. Obviously, if you have white LEDs, they're inherently going to be less efficient than if you have a lot of red LEDs. How are these measured? And this is a common question. They say they have an LED fixture. Can they measure how efficient it is? The simple answer is no. Individuals can't do it unless they have the equipment and the expertise to do so. This is an integrating sphere, one that AJ Both has at Rutgers University. And so it's basically a device where you put the fixture inside and it can measure the total number of photons emitted from that light source. And then it also, then there's a way to calculate how much energy is consumed to emit that amount of light. So it requires some expensive equipment and expertise to be able to determine what the efficacy of a fixture is. And therefore, large companies probably have the equipment to do this, but other companies may send their fixtures to testing labs to get this sort of data. So to determine the efficacy of a fixture, it's basically taking the total amount of radiation emitted from the fixture in terms of micromoles per second, divided by the amount of energy consumed to emit that electricity, to emit that light. So if you have 600 micromoles and it took 300 watts of energy, which is equivalent to joules per second, you do the division and you can see the efficacy of the fixture would be 2 micromoles per joule. And of course, the higher the value, the more efficient the fixture. I spent a fair amount of time looking for examples of fixtures that were primarily for indoor lighting applications. This is a small sampling of what's available, and I just chose ones in which I could readily find the information. Some of these are listed as DLC qualified fixtures. I'll mention what that means in a minute. In other cases, the data was available from the company websites. So I point this several points I want to make with these examples. First of all, a comparison with a T8 60 watt white fluorescent. You can see emits only 48 micromoles per second, so quite a low intensity. And the photon efficacy is about 0.8 micromoles per joule. These other fixtures are on the market today. You can see that they range from about 2.2 to 3.0. So we're talking 3 times, even coming close to 4 times more efficient than the fluorescent fixture. The amount of light emitted from the fixture varies widely. You can see some of these emit maybe twice as much as a T8 fluorescent, whereas others might emit 10 times as much. In fact, some of these would be even more than that. So obviously it'll depend upon the fixture, and that's why you can't just say, well, when someone asks, well, how many fixtures do I need? It's going to depend upon the fixture itself. The efficacies vary quite a bit, as I said. A lot of it is influenced by what is the composure of the the fixture in terms of what are the LEDs. So fixtures that typically have the highest values, like a 3.0, typically have mostly red and a little bit of blue. And then as you decrease the amount of red and increase the amount of white or blue, you're going to see decreases in the efficiencies, just because, as I showed in a previous slide, those LEDs are less efficient. So some of the lowest efficiency fixtures will be primarily white LEDs with a little bit of red. I'll take questions here toward the end, okay? There will be time for that. So just a few more slides. One, as I mentioned, the Design Lights Consortium, or the DLC. You can see their website address there at the bottom. They're a non-for-profit organization, as you can see in their mission statement here, whose mission is to achieve energy optimization, establish criteria for fixtures that can be considered energy efficient. These are some of the DLC technical requirements that are currently in place. This is version 1.2. The ones that people tend to focus on the most are the photon efficacy of at least 1.9. The photosynthetic photon flux maintenance, or Q90, should be at least 36,000 hours, meaning that the intensity of the light should not diminish more than 10% until the fixtures have been used for at least 36,000 hours. You can see the lifetime of the drivers and fans, if present, should be at least 50,000 hours, and they should have at least a 5-year warranty. They should also be safety certified by an appropriate organization. I will note that the DLC is proposing to slightly increase the standards, and the current plan is to increase the value of the efficacy value from 1.9 to 2.1 micromoles in January of 2021. For more information on lighting, we do a lot of research on lighting and try to publish articles not only for academics but for growers. You can see the website here at Michigan State for lighting, or as well as other topics that may be of interest. And then last but not least, as Cherry mentioned, we have our first annual OptimIA meeting that is planned for Wednesday, July 29th, 2020. We're planning to begin at 1 p.m. Eastern time. It should last about 3 hours long. It'll be via Zoom, so given travel restrictions, we will be meeting online. There will be no cost to participate, so that would be an advantage of meeting online. We are working on the agenda. We hope to have it finalized by the end of this week, and we will post that when available on the scri-optimia.org website, where you can look at it, and then we will have a link to register. We will require registration in advance, just like we are for participating in these Ag Science Cafes. So with that, I'd like to thank everyone. I saw a lot of activity, but I wasn't able to read what the comments or questions were. Hopefully not too many of them were revolving around not being able to hear me.